All right, this is the lecture for Chapter 26, Development and Inheritance. I'm going to concentrate on just certain areas of the material in this chapter and skip over some. Development is a gradual modification of the anatomical structures and physiological characteristics. This occurs from fertilization to maturity. It covers one cell to an individual with trillions of organized cells. It's really quite remarkable. Embryology is the term for the study of embryological development. In the first 2 months of pregnancy, or gestation as it's called, is considered the embryo or embryological period. And from the 9th week to birth is fetal development. Prenatal development is divided into 3 sections called trimesters. And the first trimester is when there's early embryological and fetal development. The rudiments, or the basic groundwork for all major organ systems, appear in the first trimester. In the second trimester, that's when those organs and organ systems develop further. They're nearing completion by the end of the 6th month. Body shape, proportions change, but by the end of the second trimester, the fetus looks distinctively human. The third trimester is where the largest gain in fetal weight occurs. Most of the organ systems become fully functional, and an infant born in the last 1 to 2 months has a reasonable chance of survival. And I would say that, in fact, being in the right place with our current state of technology, infants born earlier than that also have a reasonable chance of survival. In fact, I think they say now that from about 24 to 26 weeks of development, and they're 40 weeks in a gestation period, that some infants can survive. Postnatal development begins at birth and continues through maturity. Alright, so how this all comes about, we go all the way back to fertilization. And that is when there is a fusion of 2 haploid meaning they have just 23 chromosomes, not pairs, but 23 chromosomes. And you'll recall, these are called gametes. But when these 2 haploid gametes fuse, that produces a zygote, and that is a cell with 46 chromosomes. Generally, this occurs in the uterine tube ampulla or isthmus at about 24 hours after ovulation. Talk about being one of a kind. 200 million spermatozoa are commonly deposited in the vagina at intercourse. Of that 200 million, about 10,000 will enter the correct uterine tube. In other words, the tube where the egg is. Of that 10,000, less than 100 will reach the isthmus of the tube. And yes, only one sperm fertilizes the egg, but it requires the help of many others to help it gain access. So, at ovulation, you recall the oocyte leaves the ovary, enters the uterine tube via the infundibulum by the help of the fimbria. There, fertilization and oocyte activation occurs. The acrosomal enzymes from the multiple sperm create gaps in the corona radiata. And the book illustrates this quite well on page 946. Let's look at it here. You see, this is the oocyte at ovulation. So it is a secondary oocyte and a first polar body. These cells around it are the corona radiata. At fertilization, it requires the enzymes from multiple sperms to create gaps in that corona radiata to allow that one single sperm in. And as soon as it enters, a change occurs in the membrane, and no further sperms can get in. The oocyte then goes on to complete meiosis, and a pronucleus formation begins. You see over here in this stage you had the chromosomes, 23 chromosomes. In this stage they start to form the pronucleus. The next step, you see spindle formation begin. The nucleus of the fertilizing spermatozoon forms the male pronucleus. Then the male pronucleus and female pronucleus combine in what's called amphimixis. And amphimixis is when the two pronuclei fuse. And at this point, the cell is called a zygote. It is now 23 pairs of chromosomes and no longer 23, two sets of 23 individual chromosomes, which is called a haploid. Then step 6, cytokinesis begins, and human development is off and running. The stage called cleavage is when the zygote cytoplasm divides into numerous blastomeres, and this is called a pre-embryo. It lasts about 7 days. During this time, it's moving down the uterine tubes toward the uterus, and after 3 days, the solid ball of cells is called a morula. After 5 days, that blastomere becomes a hollow ball of cells called a blastocyst. So it goes from one cell to a solid ball of cells, the morula, to a hollow ball of cells, the blastocyst. Once the blastocyst enters the body of the uterus in the uterine cavity, it will then attach to the wall of the uterus in what's called implantation. This occurs in about 7 to 10 days. So the blastocyst attaches to the endometrium. The outer layer of the cells provide nutrients to developing embryo. That's called the trophoblast, and the inner mass of cells actually form the embryo. A amniotic cavity develops as the inner cell mass separates from the trophoblast. Finger-like villi extensions go into the endometrium, and this is going to start to deliver nutrients to the inner cell mass. Starting with day 9, process called gastrulation, when the inner cell mass organizes into 2 layers called a blastodisc. There's a superficial layer and a deeper layer. The deeper layer forms what's called the yolk sac and a second extraamniotic membrane. At this point, the yolk sac is the primary nutrient source. As gastrulation proceeds, the three embryonic layers form, also called germ layers, and this is ectoderm, endoderm, and mesoderm. And if you look at this in your book on page 951, you see which what features each of these layers produces. For instance, the ectoderm produces the integumentary system, the skeletal system, nervous system, endocrine system, respiratory system, digestive system. Certain parts of those systems. The mesoderm contributes other parts to those systems, and same with the endoderm. Okay, gestational trophoblastic neoplasia. Many aspects of the trophoblast are similar to cancerous cells. They have repeated nuclear divisions, they're extensive and rapid growing, they invade and spread into tissues. In about 0.1% of pregnancies, this trophoblast behaves like a tumor. And about 20% of those will metastasize to other tissues. And this is, of course, not a normal thing, and prompt surgical removal is necessary if that does occur. So I already mentioned the yolk sac. Another extra embryonic membrane is called the amnion. And this is where amniotic fluid forms, and that helps to cushion the developing embryo and fetus. There's something called the allantois, which begins as an outpocket of endoderm, and this will later form the urinary bladder. A membrane called the chorion is from the mesoderm and trophoblast near the allantois, and this is what will form the placenta, and once it's formed, becomes the primary support for the embryo, providing oxygen and nutrients in exchange for carbon waste products. So here you see formation of the allantois. Here next to the yolk sac you have the amniotic cavity forming, and the embryo itself is here. As you go down to week 5, see further development. It is about week 3 where the head fold starts. Now in these early weeks, they don't have features that are easily recognizable as in later stages of development. And at this stage, they're referred to as folds because as you look at pictures in your book, page 950 is one, 958, number 1 is another very good illustration that they basically look like folds or creases. It is at week 3 that the embryonic heart starts beating. Chorionic villi is formed. At about week 5, the umbilical stalk forms from the body and yolk stalks, and this will become the umbilical cord. At week 10, the embryo is now a fetus. The fetus is connected to the placenta by the umbilical cord, and the cord contains portion of the allantois blood vessels and remnants of the yolk stalk. The amniotic cavity now fills the uterine cavity. It's full of amniotic fluid to provide protection and support for the developing fetus. The placenta, this is beginning on page 956, has what's called chorionic villi, and this is for exchange with maternal blood. The placenta is connected to has two umbilical arteries which bring deoxygenated blood with fetal waste to the placenta. So these are umbilical arteries, so they're going from the baby to the mother, so they're carrying deoxygenated blood. The umbilical vein is returning oxygenated blood with nutrients from the placenta to the fetus. So here's a difference from normal circulation in someone that's already been born. Normally as you know, normally arteries carry oxygenated blood and veins carry deoxygenated blood, but in fetal circulation it's different. So in this picture you have a representation of the placenta with the chorionic villi embedded into the endometrium of the uterus. And if you take a section of that and blow it up here, you see the chorionic villi again. This is an area filled with maternal blood. So nutrients and gases exchange across this space. The baby actually has its own blood supply. The baby's and mother's blood does not mix. Alright, if you take a section of this chorionic villi and look at it in cross section, you see the fetal blood vessels here, the embryonic connective tissue, syncytial trophoblast, and the area filled with maternal blood is here on the outside, again, which corresponds to this dark blue here. The placenta produces certain hormones, one of which is the human chorionic gonadotropin, or HCG. Functionally resembles LH in maintaining the corpus luteum, so the uterine lining is also maintained. And this occurs for 3 to 4 months. The placenta is then formed and secretes estrogen and progesterone to maintain the uterine lining. It is HCG, as you may know, that is used as an indicator of pregnancy in tests, both in urine and in blood. Another hormone produced by the placenta is called human placental lactogen, HPL, and this activates the mammary glands, along with certain other hormones. Relaxin increases the flexibility of the pubic symphysis during birth. As you recall, the pubic symphysis does not have a bony fusion, but there is a very dense connective tissue between the two pubic bones, and that normally holds it very rigid, but under the influence of relaxin, that connective tissue becomes more flexible, allowing some movement of the pelvis bones during birth. Relaxin also causes dilation of the cervix and delays labor contractions until late in pregnancy. Progesterone and estrogens help maintain the uterine lining in early pregnancy, and in late pregnancy an increase in estrogen plays a role in labor stimulation and delivery. So this is the end of part 1. Please go to part 2.